Yes, it's same that you have to cut open the uh, and then put it into something and then perform a test. Yeah, similar. It's similar. Okay, go through this one and then tell me what we have to do in the question. Okay. Okay. So what they're saying cooking fruits and vegetables in water reduce their vitamin C content. Okay, the vitamin C is transferred from the food and vegetables to the water by diffusion. So DCPIP can be used to estimate the concentration of vitamin C. So what they have investigated, they took a piece of foil and wrap it around the beaker. Why do you think they have done so? They wrap the beaker with foil. Okay, good. Then a pieces of citrus fruit was put into a beaker. Hot water was added to maintain the temperature, whatever the temperature is, because we have to check it for like cooking fruits and vegetables. A stop clock was started, and 1 cubic centimeter sample of the liquid in the beaker was taken every minute for 5 minutes. They have taken five readings. Now, a syringe was filled with DCPIP, and the volume was taken 5 cubic centimeter. If there is any air bubble, you have to remove the air bubble because they might affect the result. So one drop of blue DCPAP was added to the test tube containing the first sample. Okay, now we have added a drop wise. The test tube was shaken for 5 seconds to mix the content, and the color was observed. Okay, what would be color if vitamin C is present? So DCPAP is blue in color. What would be its final color if the test is positive? It turns colorless. Okay. And if it has more vitamin C, more the quantity of vitamin C, faster the color will disappear. Okay. Okay, then step 7 and 9 was repeated, and the student observed these colors. Okay. Volume of DCPIP remaining in the syringe and volume of DCPIP added. So, wait. There is a total volume of 5. Okay. Let's say in syringe they have taken 5 cubic centimeter. Sorry. Oh ho, haven't chosen a pen. Just give me a minute. Okay. The total volume was taken 5 cubic centimeter. Out of this 5, we have added 1.4 to the test tube 1, let's say, after 1 minute. So how much is left? 3.6. 5 minus 1.4 is okay. Or you can do it the other way, that if 3.6 is remained or left in the syringe, then how much is added to the test tube? Then 5 minus 3.6. So in yes, 5 minus 3.6 is 1.4. Subtracted. Yes, 1.6. 5 minus 1.6. So the answer is yes. Okay, here starting volume in the syringe is 5. Volume of 1.6. So answer is 3.4 cubic centimeter is already written. Okay. Prepare a table. Make a table of it. Okay. Is 5. Is 5. No, it's 5. We are adding we are adding drops after every minute, right? And we have to do it for 5 minutes. Here is the step. Here it's written. Yes, yes. So prepare a table and record your result. Yes. What happened? Okay. Just make a table. Make a continuous table. Okay. So our heading should be, what is the heading over here? Volume. Okay. DCPIP. Let's say volume of DCPIP added. Okay, we should write it here. I don't know what's the issue with my pen now. It's lagging. Time, and the unit is minute, and volume of DCPIP added, and the unit is cubic centimeter. Okay, the unit should be inside box. I have written it outside the box, but it should be inside the box. Make a bigger box or do or make your font smaller. Okay, let's say 1 minute, 2 minute. Oh, sorry, I'm writing minute again. Wait, one, two. You don't have to rewrite the minute unit over here. Two, three, four, and five, and the values are 1.4, 2.2, 2.8, 1.4, 2.2. DCPIP added 3.2. No, 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 no. Here they have written something. Prepare a table and record the result shown in figure 5.1 and your answer to this. Where is this value written? Here. Okay. So we have to use this table, not the other one. We have to use this column. Okay. What is your conclusion? What can you be like? What is your conclusion? Okay. Okay. Wait. Wait. What have you told? As the time increases. Okay. This is your observation. Okay. This is not conclusion. This is your observation that. Wait. No. These are two different things. As the time increases, you have observed that when it comes to 5 minutes, the volume more DCPIP is added. Is it so? More volume of DCPIP is added. Okay. So we have what from what point we have started? We have started from cooking fruits and vegetables. That's why we have added hot water, right? So we have to relate it with that. That would be your conclusion. More. Okay. Let's say if a longer, if what should we write? If a longer cooking time is allowed more vitamin C would diffuse out because you have used more DCPIP. Okay, okay, wait. We have said that what from where we have started. Let's move to the first point. It's written in the very first sentence that cooking moves the vitamin C into the solution. Okay, this is the like simple crux of the first sentence. Okay, now we are adding DCPIP to check whether vitamin C diffuses in or not, in water or not. From this, from the cube, it should move into the water. So we have observed that as the cooking continues till 5 minutes, okay, let's move to cooking because we have used hot water. It means we are cooking the sample. So if we are giving more time for cooking, more DCPIP is used. It means more vitamin C is present in the solution. Okay? That's why we are writing if a longer cooking time is allowed, more vitamin C would diffuse out of your cube. Yes. And what sample we have used? We have used citrus fruit in the start. Okay. Everything is given in the question. Cooking is given in the question. Citrus fruit is also given in the question. So we have to use the same words as are used by the examiner, the paper setter. Okay, where is citrus fruit written? I've read it. Okay, here. Okay, that's why we are using these words. Okay, what is the independent variable? What are you changing? Wait, what that is your effect. That is dependent. What are you changing? You have made a table. Time, exactly. Time is independent while the volume of DCPIP added is dependent. Yes, yes, yes. That we are changing. Yeah. Exactly. We are taking reading after every 1 minute. It's meaning we are changing time. 1 minute, 2 minute, 3 minute, 4 minute, and 5 minutes. Okay. Explain why it is important to remove the air bubbles from the syringe. I guess I have left one question. More reliable result. Good. Or more accurate result. Okay. What is the purpose of wrapping the beaker? We have discussed this. To prevent heat loss. We discussed this already. Okay. Next one is plan an investigation to determine the effect of light intensity on photosynthesis in an aquatic plant. And I've told you to do these practicals from the book. It's a plant nutrition. Have you gone through those topics? Okay. Hmm. Yes, yes. And we have to tell the method for like how do we change it. We move the lamp towards the beaker or away from the beaker to change the light intensity. Okay. So to in order to investigate the effect of light intensity of photosynthesis, we okay, 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 done. Let's move to the next one then. Now, in this diagram, there were lots of mistakes by students. First is the shape should be correct, and these number of ovules, these should also be correct. There should be 6 seeds, okay, and then there are two openings without seeds. Here there are no seeds. Eight. Exactly, we have to shape it. 8. Shade, not shade, shape it. Don't shade it. We have to make 8 and then 6 seeds and 2 without seeds. Simple. Exactly. Exactly. Okay. Then magnification is given. Okay. When it's written 3 significant figures, then it should be in 3 significant figures. Okay. Mean including the decimal. It should be like 3, not more than that. Be very careful what they have asked. If two decimal places are written, it means that after decimal you can write two figures, two numbers. Okay. Okay. Plants can make starch and proteins and fats. What is the test? Name the reagent that can be used to test sample of plant tissue for starch. Iodine solution. Barrier. Okay. Iodine solution. Barrier solution. Okay. How do we test a plant tissue for fats? You have to give a complete method. Yes, yes, yes. Add ethanol and then we shake it to mix it up, and then we add water and shake it. And because they haven't asked about the end result would be ethanol emulsion. Okay. It's better to write ethanol emulsion. Okay. Keep in mind you have to add both, first ethanol and then water. Okay, the next thing is the enzyme pectinase is used to produce apple juice. Pectinase break down the cell wall in plant tissue. This increase the volume of juices that can be extracted from the apple. This is also part of your biotechnology chapter, right? Okay, then 7 samples are taken. It's written the 7 samples are taken, and these samples were put into different beakers, and then we add the same pectinase amount, that is 50 cubic centimeter, to 6 of the beakers, not to the seventh one. Okay, the content of each beaker were okay, then there are kept for 30 minutes at 37 degrees celsius. The contents of each beaker were filtered through filter paper and liquid was collected. The volume is then recorded to calculate whether how much juice is produced. What is the dependent variable? Independent variable is definitely okay. Dependent is okay. Independent, dependent mean your result. What is your result? Wait, look at the last 2 points. Volume of liquid. Yes, so we are measuring this. Look, it's written. Okay, that we started the experiment. Okay, we this is the independent thing. This is independent, and this one is dependent. The thing that we change, we are changing the masses. Okay, state two variables that should be kept constant. Okay, it's written in the what is given in the investigation. Temperature, 37 degrees Celsius, good. Temperature, okay. There is one more thing written. I was about to mention that, then I like didn't. What else is kept constant? What is added like content? No, there is something else that we have added in a particular amount. Okay. Volume of pectinase. Oh, it's 50 cubic centimeter was added to. Wait, volume. I'm not talking about concentration. I'm talking about volume. It's volume of pectinase, which is 50 cubic centimeter. Here students are got their marks low. Okay, identify a hazard. Identify a hazard. You are using enzyme. You are cutting something. So what can be hazard? Cutting, chopping. So what can be done to prevent any injury? Okay. What? Wear gloves or cut away from the body. This is very important. In the opposite direction. Okay, the student did not repeat the investigation and collected only one set of result. Why it is better to repeat the experiment? Good. To identify anomalous result. Okay. The next question is the seven beakers of chopped apples did not contain the pectinase enzyme. We have added pectinase to only six. Why haven't we added? Okay. What should have been added in the last one to make it control? That we have added to the six beakers. Now, what should be added in the last beaker instead of pectinase? Water. We can add water. Okay. We discussed it earlier. What is control? Okay, wait. We'll discuss it later. Let's leave it here. We can add water or we can denature the enzyme, denatured pectinase. Do tell me that we need explanation on this question. Okay. Then the readings are given. Use the information to calculate the volume of liquid collected. How much liquid is collected? Okay. We have a sample of 150 gram. Okay, this is a total. Okay. And we have added 0.8% of pectinase solution. 0.8% of pectinase solution. Okay. So the overall reading is this. We have to use this one. Okay. What is 0.92 of 150? 0.92% of 150. Wait, its percent is also written. Okay. So what would be volume? It would be 138. You have to multiply it with 100. Okay, wait. Let's just erase this. The total volume is 150. 150 is the mass of chopped apples. And we get, let's say, 92% of this one as a juice, right? So what is 92% of 150? Hmm. 92% of 150 is what? 138. And the unit should be because it's a liquid. Okay. Then we have to plot a graph. Okay, what is the relationship of volume of liquid collected and the concentration of pectinase? Okay, here is a table. What can you detect? Okay, conclusion. as the concentration of pectinase increases. This is the conclusion. Okay, 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 okay. Now these are two different cases. Okay, wait. Let's move to this one. Where is the conclusion? Okay. So you have to conclude the whole result that you have given it time to get boiled or to get cooked. That's why we have added that if a longer cooking time is allowed, then vitamin C would diffuse out more. Okay? Okay. The longer cooking time, because this was given in the question. Now, the question over here is we have added, wait, where is the? We have added the pectinase. So we are not changing anything. There they have mentioned cooking. Here they have mentioned pectinase. So that's why we are using this word. That if there is like more concentration of pectinase, what is the result? There, the volume of liquid would be collected more. Wait, have you? Okay, here it's written describe the relationship. Now the question is different. It's not a conclusion, it's a relationship. Yes. It's about x-axis and y-axis. It's about x-axis and y-axis. Okay, what would you take at x-axis and what would you take at y-axis? Okay, good, good. So what would be your units? Pectinase. Cubic centimeter? Oh, volume of pectinase or volume of juice? Liquid collected. Okay, then I should correct it. Volume of liquid collected. Liquid collected. Whatever is written in the table, you have to use that word. Okay? And here we have to use the word. Okay, it's written per gram of chopped apples, so we have to mention that. Per gram of chopped apples. We have to write this whole. Okay? And then percentage concentration of pectinase solution. Percentage concentration of pectinase solution. Okay. What should be your scale? It should be 0 0.2, 0 0.4, 0 0.6, 0 0.8 and 1. Right? Okay. And what would be other scale? The value is still 0 0.92. What can be done? 0 0.92 is from 0.69 to 0 0.92. Yes, you can start from 0 0.5, you can start from 0 0.9. 0 0.60, okay. 0 0.80, 1.0. Okay, okay, wait. Let's make it bigger. Instead of 0 0.60, right here 0 0.70, 0 0.80, 0 0.90. Okay, okay, and then 1.2. Done? Okay. Now you have to make, okay, attempt this question related to plan and investigation and then send me the answer.